All right, welcome everybody. It's great to see you everyone here today. Today is when we're going to start our journey into RNA, right? So the first few workshops have covered DNA analysis and R and kind of looking at all the different things we can do with it, from lollipops to phylogenetic trees, right? I think we did a pretty good job of covering everything. But it's time to move on now to RNA, right? Which is the pretty much the most popular thing people are doing nowadays. Everyone has an RNA set they're working with, especially moving the transcriptomics and obviously trying to couple it with integrated, imaging pathology, things like that. This is very cutting edge. But just to move into the start of, we just kind of start at scratch, right? Bulk RNA seq, and then we'll move on to those more advanced methods. But let's just start from scratch here. And one thing I want to talk about today is the difference between DNA and RNA. So I was talking to a friend about this, and really, DNA is an engineering thing, right? It's like becoming a mechanical engineer or chemical engineer. There's pretty obvious settings and parameters and guidelines for following it. It's pretty easy logically to see how reads fall as alt reads or normal reads, and it's not easy data to work with. But compared to RNA, it's more of an engineering battle, where RNA is more of an artistic. I don't want to say subjective because I want to give more value to it. It's an art form. It's like being a painter versus an engineer, right? So you have to kind of develop this intuition working with RNA because there's just so many ways of different parameters, different batch corrections, different changes that you really have to understand. It's just very integral to understand both the mathematical attributes to these methodologies and also the biology, because that's the main thing. A lot of the people that computer scientists work with this, they don't understand the biology and they get results. They look good, but they're not real. So just always know you need to keep cycling back around and kind of a flywheel to make these things work with the biology. All right. Well, that being said, let's dive in. Welcome to the world of RNA, right? I know you guys pretty much know about this stuff, but today we're going to be talking about bulk RNA-seq. It's the most widely used technique for measuring gene expression across the entire transcriptome. And really, if you think about it, if DNA, mutations give us the blueprint, like what probably should happen, and RNA expression is the readout. Because a lot of things happen between a mutation and a transcript being produced. So we're really seeing a downstream expression of these mutations that complement the data and our understanding of the tumor, right? Um, and don't knock bulk RNA-seq. I did single cell transcriptomics for 5 years, then I got my first job, and all people wanted me to do was bulk RNA-seq. So it's still very important if you have things like mouse data. I know people that work with the xenograph mouse data. A lot of times that's going to be just bulk expression to try to find drug discovery and things. You don't need a super expensive experiments like single cell for that kind of things. And also a lot of your validations are in TCGA, different things in Portal. So don't knock bulk. Bulk is still really important, and most people still do it wrong in a lot of ways. So it's really important. You could jump on a nice tutorial with Michael Love or those guys that wrote DSeq, run through 5 lines of code and get an answer, give it to your PI, it looks good. But it's probably going to be wrong. So today we're going to show you how to do it right and some of the things and pitfalls that can fall in between. So why do we study gene expression in cancer? Well, you guys know, to look for things, to understand, expression levels can give us understanding of oncogenic pathways that are active, not just mutated, but actually turned on and being expressed, right? Very important thing to try to figure out are the different different cell types, cell states, transcriptional programs that define molecular subtypes, and really look at combining this back to the clinical data to see how it predicts patient survival. And really the clinical impact is enormous with RNA-seq. You can do molecular subtyping, you can do immunotherapy prediction, drug sensitivity, prognosis. It's just extremely important, and there's a reason why people like to work with it so much. So let's dive into our cancer model. What will we use today? Well, we're going to be using LUAD, which is a great, one of the most extensively profiled cancers in TCGA, which is what we'll work with today. And it's just a great model to be able to look for a lot of these things as we move on to the downstream. It's really cool immunogenic tumor with a lot of macrophage infiltration. It's a very interesting tumor, I think. So for this sample, We're going to pull in just 500 tumors with 59 matched normals, which is really good distribution there. There's 3 main molecular subtypes. We'll see if we see them. We see key drivers in KRAS, EGFR, TP53, STK11, and there's a clinical challenge with survival. So survival is going to be important to look at, and the immune landscape is heterogeneous. So they have immune hot tumors, immune cold tumors, and it really matters for treatment. Depending on these the level of immunogenicity between different populations, right? So what we're going to do here is really I haven't looked at this data. I've worked with LUAD data before, but I haven't really delved into TCGA LUAD anytime soon. So just think about this as we're just doing this together. We have some bulk RNA-seq data, right? What can we do with it? And let me show you. Let's work through it and see what we get. I don't know what it'll be. Let's just find out, and we'll talk about it. So we're going to, download our data, do some quality control, do some different normalization strategies, apply our dimension reduction and clustering, do our D-seq analysis, visualizations, we'll do some functional enrichment analysis, NMF, and finally some immune cell type deconvolution and survival. So we're going to be covering a lot today because I want to show you everything that you can do with this. Bulk RNA-seq is just going to be one workshop because we want to spend more time on single cell. So I'm going to tell you everything you need to know about bulk today. So let's jump in. So first, obviously, you need to install your R packages to get going. There's usually 3 ways to do this: through CRAN, as you guys know, R buy-in conductors, you guys know, and also through GitHubs. If you install DevTools, you can do things like pull in this immunocom or deconvolution package from GitHub, which we will be using. So definitely run that to pull that in. And then we go ahead and load all our packages in with the annotations that you guys can kind of check out later. And we'll jump into actually downloading our real data. Well, if you've been following along on my different lessons, we've done this a million times, where we're just going to use our TCGA biolinks to pull this in. So first, we do our GDC query, where we just pull in our LUAD, transcription profiling. We want gene expression quantification. We want star counts, very important. We want raw counts. Anything other than raw counts will not work for us. So we just want the star counts. That's raw counts. And we want primary and solid. And then we download that query and turn it into a summarized object. And a summarized experiment object. You've seen them before. It's basically just a container for the assay data and metadata. It's just really a superpowered list that keeps everything in power. And we'll see how single cell uses something called a surrogate object for that as well too. But it's just a way to organize everything and keep it together. So SE LUAD, what is SE LUAD? We can take a second and take a look at this. Where are you? SCC LUAD. So right here, and we see how it just has some of the metadata essays, things we have in there. And as we add more. As we do more analysis, we'll add things back to this object to be able to keep that all together in one spot for us. All right, so we have that, and we see we have 599 samples and 60,000 genes, which is to be expected to the start because you have your pseudogenes and all that. And let's just kind of take a quick peek at some of our sample metadata. Let's look at the clinical info, which comes with it. In this aspect, I believe is loaded in there with it, so sweet. That is a super help. Sometimes we have to wrangle that. Let's just take a look at that really quick and just spread that out. And we see we have our sample types, race, we have our stages, and we have our vital statuses, which is really good because we'll be able to do survival analysis later. And the genes too, we have our gene IDs and our gene names and the gene type, which is going to be very important to be able to extract out. And then we kind of just pull out our raw counts from our summarized object. We do unstranded, assay, and we have our raw counts, which is extremely, a pretty sparse matrix where you see just our different counts at different spots, right? So we have our genes on the left, and our columns are our actual bulk samples, right? Now let's just do a little bit more cleaning on the clinical data here, and we see that we just kind of spread out our tumors between 540 primaries and 49 normal. So we just kind of spread those out a little bit, and just looking at the stage distribution one time. This is just exploratory analysis to get a feel for the data. And we see that everything, a lot of the higher stages are more representative, but they're just kind of random. There's a little bit of distribution between those. So it's a little bit unbalanced. There's far more tumor samples, but TCGA is like that. And DC handles that well, so we'll give it a shot and do the best we can. It's all you can do with TCGA is the best you can. So the first thing we're going to do is extract out, let's look at the gene annotations. They're not all protein coding, so we take a look at all these, and we see we have our pseudogenes, small nuclear, RNA, all different kinds of, specialized RNA, pseudogenes, long coding RNA. But for us, we just want to look at the protein coding genes for expression today. And there could be reasons why you look at other ones too, but for the sake of the lesson, to simplify, let's just take those right now to use those. So now that we have our raw counts, we have our genes, right? Why can't we just go ahead and go forward? Well, the problem is, there's several problems. We have a library size problem, right? Some samples have more total reads than others, but not because of the expression, just because of something that went on in the experiment, a different sequencer, a different day, and we need to normalize for these things, right? Just because you have more expression, you have more reads, doesn't mean you necessarily have more expression, right? And that's where UMIs are actually really important later on in single cell data. There's also gene length. Longer genes are just going to capture more fragments, so it looks like you have more hits there, but they're not really real hits, or they're duplicate hits, you know what I mean? And then you have composition bias. Sometimes you have a few very highly expressed genes that eat up all the capacity, and there's not any reads left. So the only reads left go to the people that are left, and it makes them look like they're lower when they're not. So we need to normalize the control for all these different things. And for library size, things like dSeq2 and TMM normalization work really well for that. For gene length, we want to do more TPM, FKPM, and for composition bias, once again, dSeq and TM are the best for that too. We'll talk more about all these methods here. First, let's just visualize our count distributions. So I'm just taking basically the library size per sample. Samples are ranked across here by total counts. And you can see if you look at these guys, there's a huge difference in the amount of reads per these samples, right? Does it mean this sample actually is expressed like freaking 80 times more or whatever? No, that's not correct at all. It just means that for whatever reason, this sample has a higher library size, right? So we need to normalize for this. Perfect example of what we need to do. And if we look at the histogram of this data, it also has a Stark thing here. We have this kind of negative binomial on the side here to be expected with this bimodal pattern. It's very common in bulk RNA-seq data. You can see here on the left, this spike, it represents genes that are, off or barely detected. They're noisy, they're tissue irrelevant, or they're very low expressed. We don't really want those genes there, right? But on the right, we have this hump here. That is our most expressed genes, our housekeeping genes, our structural proteins, our metabolic enzymes. These are the kind of things we want to find. And then also these kind of in-between states are also very important too. So that gives us an idea of the distribution of our counts. Let's talk about what we can do for the quality control for this, right? So QC is so important in RNA. I cannot tell you. You can mess up your QC in DNA and still end up okay. But if you mess up your QC in RNA, you might as well just give up, especially in single cell. Because bad samples ruin everything downstream, right? And one thing about RNA, they're hard experiments to do. So a lot of time you have a lot of noise and a lot of issues with them. So it makes sense you need to understand how to quality control for these. So what can we do? We can look at two main levels: a gene level filtering, so which genes do we keep, and sample level, which samples we want to keep, right? So first with genes, many genes in the genome are not expressed at all, or expressed at so low levels they're just kind of useless, right? And they just kind of add noise without adding signal. So what can we do to get rid of these? So first step is going to be grabbing just the protein coding genes, like we talked about before. And then we're going to remove low expressed genes. And by doing this, kind of a rule of thumb, we want to keep genes with at least 10 counts and at least n samples, where n is the size of the smallest comparison group. So in this instance, that's 59 or 59 normal, because we want to compare normal versus tumor. This is a very important concept. It's really important that if a gene is not detected in as many samples as the smallest group, we have no statistical power to do differential expression between those two things. Okay, think about it that way. So we're setting up an experiment to do differential expression in a very simplistic way to look at normal versus tumor. So the smallest out of those two populations is normal, 59. If we had, let's say we wanted to do something else where we split the tumor into three populations, right? And they all had, I don't know, 20 in each one of them, right? 20 samples in each one of them. I'm making this up, right? It's not the 500 or whatever. Let's say let's say that there's 20 in each one, right? We split it up, to 20 different samples. We're looking clusters. The point is you have smaller clusters, right? That have smaller numbers than 59. So in that case, we would be doing comparisons between things of smaller values, right? So then we would actually be able to pull in that smaller number, and now we could factor, it would be 10 counts in at least 20 samples. So the point is, it matters how you set your experiment up. The way you want to do differential expression, the way you want to compare different clusters or samples. That's the way you set up this n value, and that's how you go for that calculation. Long story short, right? So we basically did that. We took our protein coding and took out the ones that only had 10 counts in those. And basically, we lost like 40,000 genes for protein coding filter to be expected. And we lost about 3,000 extra knocking out those kind of low expression genes. So now we have 16,911 cells with our smallest group of 59 samples, right? And 500 left of the tumor. And let's kind of look at this on the plot. Once again, we talked about that 59 cutoff here. And we're basically seeing genes over here on the left where they have a large peak of low expressed genes, mostly noise, where we have kind of a broad peak of reliably detected genes here. So we need to make a cutoff somewhere here, and 59 is the right statistically place to put it. So we get rid of all these kind of bad genes or low expressed genes and really focusing on these guys and this tail for intermediate. So that's when we kind of check everything out and see visually that our gene level filtering is kind of making sense. Now, what about sample level? Usually sample level are kind of outlier samples that usually have problems with things like mitochondrial content or ribosomal content, degraded RNA, dying cells, the number of counts, things like that. So you can look at these in a lot of different ways, and a lot of those are tackled more in single cell. So one of the things we really look at just in a simplistic way is just the library size of these samples. Are they super big, super low, and what's going on with that? So if we do a library size after filtering, and we set a filter of 3 standard deviations below the mean. So we basically set a median value of reads, and we say, hey, let's set a low threshold for 3 standard deviations below that, because there's probably not enough counts there. There's just not enough counts. So those should have a low threshold. So I set up this low threshold here, and then filter by that low threshold and just get rid of those low cells that are super low. And here we found there was only run that low. So that means this is good news. This sequencing is fairly consistent across samples. Nothing really jumping out. So we could remove that one sample. You could also make the standard deviation lower, 2 standard if you want to be tighter. That's just kind of more of that artistic way of organizing these things. So if you want to look and kind of visualize where that cell is, you might have thought it would be down here or over here. So those are different sample outliers. You can use things like dbScan or different partitioning methods to find things that are outliers like that you might think shouldn't be there. So that's another whole level of a whole other tutorial we have to do that. But just to kind of visually see this, we see it's over here in the primary tumor, kind of well clustered. So taking this out is not a clustering artifact. This is merely a library size artifact. So we are going to go ahead and get rid of those because everything else looks good as far as the PCA. And we'll just remove that one sample. So now we have 598. So no big deal. We'll move forward with that. Now, super important. Lots of supers. Normalization. So now that we have our clean filter matrix, we need to normalize to remove any technical biases so we can fairly compare the expression. And now this is probably one of the biggest things you can do right or wrong to mess everything up or make it work, right? So there's really 4 different ways I want to talk about today to do this. So 4 different methods, and you have DSeq2 and TMM, which are very similar, and they basically normalize for statistical testing, and they're good for they're not good for cross-gene comparisons. So the idea here is these guys mostly do a great job of correcting for compositional bias and library size by computing size factors. We'll talk about that in a minute, but basically you can compute a size factor based on the variability or variance of your genes, and then kind of use those size factors to normalize things to a similar level. And trim mean of m value is very similar to that, a different math, but very similar. And these are both used for more differential expression. So what I mean by that is it's not good for cross-gene expression. So you wouldn't want to say you wouldn't use it to say oh, is T53 the most expressed gene in this whole sample? No, you'd have to use it between samples, between clusters, between things split out, right? That's how you use those two normalizations for. It's the only time to use these. Now TPM is kind of interesting, transcripts per million. It adjusts for library size and gene length. So the first two, you don't have to worry about gene length because the same genes are in each population. If you're comparing normal versus tumor, it's basically the same genes, just different expression levels. So it doesn't matter the gene length, right? They cancel each other out, or they normalize themselves, right? But TPM, you're looking for absolute expression. So you do need to look for gene length, because if a gene is super long, it's going to look like it has more expression when it doesn't. And this is how you do absolute comparisons between genes in the same sample, the cross-gene comparisons. And this is really good for things like the deconvolution methods, things like that we'll look at later. And finally, VST. VST is a variant stabilizing transformation. It basically normalizes the medians and does a really good job of making the variance independent of the mean. And this is really good for visualizations, right? Same TPM. These bottom two are more for visualizations, clustering, dimensional reduction, things like that. They're not good for statistical measurements like DSeq and Lima, things like that. So this is kind of a good summary of those methods and why you'd use them for different things. So really, and just remember that, DSeq2 and TMM are for statistical testing. VST is for exploration. TMM is for cross-gene comparisons in one sample. And remember, raw counts are input only. Raw counts for all these. You need these to be able to do these the right way. That's why we couldn't take those counts earlier. And things like Bioporter always give you these, and you're like, come on, man, that's just not useful. Please give me the raw ones. All right, so let's just look at the data before we do any normalization, and we're going to basically just convert this to a readable plot where we have the samples left to right, and then we have the log 2 count plus ones reads, and then we have primary and tumor on here, and then the medians listed. And kind of like that library size plot I showed you earlier, you see kind of a little deal here kind of going like that, but you can really tell these are not, they're not even, they're not normalized, right? If all the boxes set across the way in the middle, then we wouldn't need to normalize. So this is a really good plot to see if things look great, then don't normalize. Remember, you never do things if you don't need to with RNA-seq data. So first, let's start with the DSeq method, probably the most popular one. Remember, it uses the median of ratio methods, and it computes a size factor for each sample that accounts for both library size and composition bias. So the way you do this to set up just your normalized counts, we did this last semester too, if you go back and look at some of our DSeq analysis, but you do DSeq data set from matrix, we put in our counts that we have from above, and we put in we have a clinical GC. This is just a data frame here, clinical CC, a data frame, and it has so basically here's a really important thing. So clinical CC is your metadata. It's going to have a column called sample type, and so you'll know normal or tumor or whatever, and that's basically your metadata column. So DSeq doesn't like the sample IDs to be in the first column. It wants them to be in the rows. So you move the column to row names here. So sample ID is moved to the row names. That's all that is. This is your clinical metadata. You're calling on the sample type for your count matrix, and that gives you your object. And with that object, we can do estimate size factors, and that gives us really our size factors. We can take a look at these guys and plot them, but this is basically your normalized counts right here. You see the size factors. You can see that as the variance and library size changes, you're going to have to add different size factors on these to balance these guys out, right? You can imagine that. And we do counts, DDS, this gets us our actual normalized counts. And if we plot these, you can see that everything has been corrected very well. The library size is pretty similar, and these means are pretty much together. And just importantly, the normal tumor samples are interleaved. That's great. And it looks like all the sample mediums align tightly. And it means that library size and compositional bias has been removed. So we're in good shape there. The second method is very similar. It's the trim mean of M values, TMM. And it's an edge R approach. So you can take the downstream of this and feed it in either edge R or lima. Lima is the more common. And it's very similar in spirit to DeepSeek's method. The math is just a little bit different. We won't go into that today, but it's very simple. And what you do here is you create this DGE list. So I have my counts QC, and then I have my clinical QC data frame with my sample type column. That gives me my DGE list. And I put that in here. I do calculate norm factors. This calculates the TNM norm factors, very similar to the estimation factors above, right? We take a look at these, and they're kind of set the same way as DeepSeek and Applied, as you can imagine. And you basically do one more step, CPM of DGE, and that gives you your final normalized count. And that's just like normalized counts. That's your final answer. And then we're just going to log to that +1 just to get our thing to show you here. And we see here, once again, that they've been normalized. They normalize pretty well here. And if you compare like the before and after, the DeepSeek and TMM look pretty similar results. So the choice between these two are more what your downstream analysis is gonna look like. So TPM, now we're changing things up a little bit. This normalizes for both library size and gene length. And this is really when we want to compare expression levels between different genes in the same sample, right? Like, is MYC expressed more higher than QD3 in this same tumor, right? Or doing deconvolution, we'll talk about later. But it should never be used for differential expression testing. Really make sure you don't do that. One of the common ways to mess up these analyses is by using the wrong normalization for the wrong thing. Because they'll give you an answer and it might look good, but it just won't be right. So to do this, this whole code block, all we're doing is finding the gene links. So you guys can go look through this, but I'm just looking at the ensemble, looking up online, grabbing the gene links, calculating the effective gene link, and then I have a vector of gene links. And it's just basically just giving me the different gene links for all the genes. And then basically to do TMM, you can think of it as really simple. You basically just divide each gene's count by its length, right? And that gives you reads per kilobase. And then you sum all these in a sample, provide a scaling factor, and then you get your transcript per millions. But think of it as just normalized it's just your reads are normalized by your gene lengths. So if some samples have more genes with longer links, they won't look bigger. So this brings that all back together, and I run that basically the TPM. Once I get that kind of set up and the way I want to, then I can put it in here and do compute TPM, and I get my TPM matrix. And then I do that +1 log 2, and that gives us our final plot here. And this is going to, on our final, but our third plot here, and this really jumps out to me. It's all jacked up. They're not equal. What's happening here? Well, the thing is, the difference is DeepSeek-2 and TMM look across all your samples, and they ask, do we actually make these comparable? Or the alignment is actually the goal, right? Where TPM does the math within just each sample on its own. So samples if they end up being aligned, that's a side effect. Remember, we're just dividing each sample by its gene length and doing some calculations. The other ones all try to align. They all work together with all these guys to try to model them and everything. Each one of these samples doesn't care about the other samples. They're just normalizing themselves. So we end up having it's a little bit more straight, right? But it's really jaggedy, and that actually is to be expected. And like I said, this is more for our cross-chain comparisons, right? Our final normalization is our VST, which is a very great method. It's used a lot. And this is for visualization. We don't want to use the raw counts or even the normalized counts because RNA-seq data is heteroscedastic. Can I talk? The variance depends on the mean, right? And those. So we got to make that independent to be able to make these plots and comparisons the best way. So we take that DDS object at the very beginning before we did a differential expression, and we do VST this time instead of our DSEG2. And then we basically grab that essay, and we have our final matrix. And just looking at the raw counts versus VST to kind of look at what we've done here, on the left here, you can see these lowly expressed genes have a ton of variance, right? And that really just messes everything up. And one thing VS2 did is kind of balance that. It brought those down to where everything is more normalized, where variance is independent of the mean, but they still have their normalized expression values. So that's works really great. And we can see here, if we plot that, it looks pretty awesome. It stabilized things really well. It really shows that it does double duty. It normalizes by aligning the medians and stabilizing variance. So this is just really awesome for downstream analysis for a lot of our methodology. So now that we've done that, now we've done a great job. We have our data, we have it filtered, we have it normalized. So we're just ready to go into the real analysis now, right? We're really rolling good. So the first step, as always, do your PCA dimensions. You want to be able to plot the best signal to show in that, right? And really, pardon me, obviously PCA shows you the major axes of variation, so we can really see what is going on. So I'm just doing PCA on the VST, obviously, right? So I grab my VST matrix, and I'm just taking the first 500 most variable genes and doing a PCA on that. And it's very similar to the plot before. It's just a little bit tighter, right, on these guys. So I feel pretty good about that. And it confirms the dominant source of variation is in the tumor versus normal tissue, which is to be expected, right? So we feel good about that. Where we do other things, like if you plot stage, we're not seeing a separation in our PCAs for that. And so you can always plot your different variables in there and see if you have batch effects, number one, and number two, see if they split off themselves. I'll really tell you if you're doing your experiments correct. But this actually makes a lot of sense, and it's something that people don't think about very often, and it's an important biological lesson that clinical staging is based on tumor size, lymph node involvement, metastasis. These are real things, right? And they're also stages based on actually looking at actual real slides. There's a human component to it. It's not doing any of this. We're just looking at the transcriptome. They don't have any of these things to compare it to, right? So for instance, like stage 1 tumors can have very different gene expression profiles, and a stage 1 tumor can look similar to a stage 3 tumor. And that's exactly why we do molecular subtyping, because it's hard to tell what's going on with just stages. But I'll just show you that not everything can be clustered if you think about it biologically. And just a quick scree plot here, for our PCA dimensions, how many we want to take. If you look at the elbow plot, it's hard to say here, but probably around 5 or so, or 6. Just to be careful. And just to kind of look at some of the heterogeneity in our data, we just look at the distance here. So taking our VST matrix, we just calculated the distance and plotted it just for kind of a sanity check. And we see that the solid normal are here. The distance is very short to be expected. And then we have different programs here, possibly of genes that are farther apart, and and ones that are closer, things like that. So we can see that there's clear structure in the data showing that they should be able to find clusters with biological meaning. And jump one more step, do the correlation, the top 500, and we're kind of just seeing a similar thing where we see obviously the normal cluster is very correlated. You see 2 anti-correlated clusters here, which makes sense too. And then also you just see these clusters of correlation and non-correlation, right? Exclusivity. So we really see that we really have a belief that we see good structure and data here as to be expected. Now, what about handling batch effects? So single cell RNA, bulk RNA, batch effects, huge problem. If you run an experiment at one location and another at another, so many different factors could go into effect and handle the way your samples are processed, sequenced, everything, and it can change the expression values. And RNA expression is notoriously sensitive to these kind of clinical parameters or research parameters. So it's very important to know if you have batches to correct for them. On the other side, if you don't have the batches or you're just not quite sure, always check the biology, because you also want to do the least batch correction possible. So it's a double-edged sword. RNA needs the most batch effects in theory, but they are punished or damaged the most by doing batch effects that aren't needed. So it's just a very fine line of recognizing these effects and then not over applying them to kill the biology itself. But let me just show you a couple of examples. So our data is, there's no batch effects in them. They've already been taken out. So we can't show that here specifically, but what we're gonna do is do some synthetic data. So what I did was I took our real data, right? And I made a new thing called clinical DC batch and just made a new column, and I simulated 3 batches. So I added just 3 random batches to our data just so you could run the code to see if it works and it's right. So you can see here, clinical batch, right here, I just added a column called batch, and I added, different batch 1, 2, 3, whatever, right? So how can you handle this, right? There's 3 different ways to do this that I think are pretty straightforward for things if you know the batches. Well, the first is DSeq2, and this is the best way. And if there's any way through DSeq2 or LIMO as well to model batch effects as part of the design formula, that's the #1 best way to handle this. You really don't want to take out the signal. You want to just put that into the design formula when it does its, modeling and estimations. So what we're going to do is when we make our matrix, we have our normal counts, our normal clinical data, but we added the batch, right? And instead of just having sample type, we've added batch now first too. So now there's 2 different variables that it's going to be working with, different covariates, right? And it will estimate the batch, it will estimate those batch effects into the data, and our final data will already be normalized for that. If you want to do it, let's say we've already done our DDS and differential expression, and we want to make and we already made our VST matrix, all right? And we're like, oh no, we didn't do batch correction. Well, you're okay. You can just use you can use remove batch effect, put in your VST matrix, and for your batch, just do the same as one up here, right? Your clinical data with its batch column. And that'll give you a VST corrected for batch effects. So if you want to do it later on downstream, you can do that. Another more aggressive way is to use combat. I don't like doing combat because it just takes out what it considers the batch effect. It actually takes out samples and it can mess things up instead of modeling it. I don't really like doing that, but which is always good to try too. Sometimes it gives the better answer, right? For that, you just run combat. We did this last semester as well. You put in your VST matrix. You basically just tell it the batch, and you have another model here where you basically show in your design signal, and that tries to hold your other signal in there as it removes the batch effect. So it tries to correct for it, but it's dangerous. And that's if you want to do it on the VST data. If you want to start with raw counts, you can check it out here. Very similar here, combat C counts, basically your column and your sample type once again to try to hold the signal. And that would give you your corrected results. So if you ran these 2 methods, doing it LIMO or DSeq is the way to go for your design formula. Or if it's too late, do your remove batch effect for VST. This is all going to give you your great data to be happy with to correct for your batch effects. So what if we don't know? What if we don't know the batch effects? That's a major problem too. Like we don't know the batch numbers, but we know there's a batch effect 100%, and we can plot it. It's there. We know it. But we don't know really where it is specifically. So what do we do? This is not a good position to be in because you don't want to overdo this with RNA. So only do it if you feel like you really, really need to do it. But there's lots of methods for this. They're getting better and better. There's a lot of deep learning methods, Python methods. I want to show you a simple one you can use in R that's been around a while. It works pretty great. Not super great, but none of them do. It's called surrogate variable analysis, and basically you're basically making these surrogate variables up to handle to represent the covariates in the data. So the first thing you do is you set up the models. The first one is by the sample type with your just clinical data. The second one, you basically just do a normal one to compare through the intercept, and then you basically estimate you as CVSeq to estimate the surrogate variables from the filtered matrix counts. This is a big step. And then once you have those variables, you just add them back. And you can see here, what you'll end up doing is putting them in your design formula. So however many you end up with, it'll be SV1, SV2, SV3, and then finally your sample type. So once again, the good news is you're putting them back into the design formula, which I said is the best way to do it for DSeq. The bad news is it's really hard to model these things well to get those surrogate variables. But anyway, that's how you can do it if you don't know how to do the batch effects, right? So now that we've corrected for batch effects, let's do our differential gradient expression. And we're going to do this with the most popular method, DSeq2. And we will do this to find genes that are significantly differentially expressed between tumor and normal tissue. And then we'll also use GLM shrinkage to really pull this, to shrink this down into a really more optimized answer here, which is really the gold standard to do this nowadays. And so what DSeq does behind the scenes is first it estimates size factors like we saw above, really our normalization. Then it estimates gene level dispersion. So this is really important. It tells you how variable each gene is. And then basically it fits to a negative binomial model to test the differences between genes, which is really good because all this genomic data is always in a negative binomial model anyway. And then we use the LFC shrink function afterward to apply APLM shrinkage to the log fold changes. And this basically pulls unreliable estimates towards zero. So it shrinks in more things with more variability that we don't have as much faith in, more to what the model is predicting. And it gives you a more reproducible effect, especially for genes with low counts or that are highly variable. So we already have our DDS. Remember, we made our DDS normalization from above. So all we're going to do is do a re-level here to make sure that the reference is solid tumor. So we tumor versus normal, right? So if you look at a volcano plot, the left will be the reference. So it's just kind of good to know, because then you'll always be wondering downstream, is it reversed, which I've done before. Then now that we have that set up, we just do DSeq on our object, really simple. And on that answer, we do the LFS shrink, and then that gives us a result. That's all we gotta do. That's what I'm saying. Like, this is all you really have to do. So that's what you're gonna see in tutorials. But think about all we've done just to get to this step. If you just ran this right away, it'd probably be wrong. Or even if it was right, you wouldn't be able to explain why. So now that we just made kind of a tidy file to look at this, and we see that we have a ton of genes here, and we'll go a little bit farther, a little bit more tidying, and we see that we have almost 13,000 genes that have a adjusted value. Remember, adjusted values are the most important thing when you're doing this kind of stuff. Differential gene expression, never look at a p-value. It's completely useless. That's a major problem you could have. Always look at the p-adjusted p-values, and sometimes they'll all be non-significant. It happens a lot, so don't be discouraged. That happens. So we have 12,000, 30,000 at below 0.05, 0.01, not much more. So a lot of these up here were also below 0.01. We start acting in the log fold change, then we start cutting out a few more, and we look at below 0.01 and above log fold change absolute value of 1, we have 4,661 cells. So those are going to be really important differential genes to be looking at here going forward. And let's just look at the top 20 differential genes. I'm just looking at these by log fold change. And you can see that they all have adjusted p-values of 0, which means they're super, super, super small. And these are the log fold changes, absolute values, I believe, yes. And what we're going to see here is, obviously these genes are very important, but what I want to stress to you, and one of the I think common problems people have at DSeq is they look at the top genes, whether it be adjusted p-value or a log fold change, and they're like happy if they're good or bad. But not the ones at the top aren't always the ones you're looking for, and there could be a biological reason other than your question why they're up there. And I think this is a perfect example of looking at that. So look at this, kind of look at the results here. We have FAM83A here, right? Which is a known Luad oncogene linked to EGFR signaling. We have this AKR1B10, which is a smoking marker associated with lung cancer. PRAME is a good immunotherapy target in lung cancer. But other things we see, kind of tumor subsets, like the TFL1's more of a tumor, the VL1 is more in tumor too. So there's just kind of a mixture. There's just really a mix results of log change results. And this is exactly why we complement single gene results with the pathway analysis going further. So, there's kind of a mixture here of tumor, immune, Luad. So it could be that the genes we cover are being shoved down because there's other biology pressing forward. So this is just our first look. This is not our final answer. This is just to get our differential genes and get a good feel for it. Just to kind of see how the model worked. We look at our plot dispersion estimates. This is our dispersion estimates, right? Dispersion over against mean normalized counts. You can see the model. So what you're seeing here, the black dots are just your normal guys. Let me just, the raw, that's right. The blue dots are the shrunken estimates, and these guys up here, the circled, are a little bit of the outliers. But what I want you to see is, as we go up to the more variable genes, they're shrunk down more toward the line. So the more available you are, the more you're shrunk toward the line. Where these guys are already kind of near the line anyway, they don't need to get shrunk as much. Because the more expression we have, the more we trust them, right? The lower the counts, the least we trust. We got to shrink them back down to what the model is saying this should be, right? So that gives, that's how you're correcting and normalizing for that stuff. And if you see a weird like dip down or some weird things going on here, or this goes up here, we know that our QC is bad. So this is a final good QC step to make sure everything looks like it's going okay. And then we're going to do a volcano plot, basically. You can do a fancy ggplot one here, or you can use the easy volcano plot one down here, where all you got to do is put in res tidy with their gene names. And we see here we have a nice plot. This ggplot code, I'd urge you to check it out. It's pretty cool that I wrote that you can handle and be very specific about things. But what we see here on the right, we see our upregulated genes for the tumor, and on the left we see our upregulated genes for the normal, or down the regulated tumor. Anyways, two ways you want to look at it here. On the upper right, we see they are upregulated in tumor. They're dominated by like top 2A, things like that. That we have our lung cancer marker, top 2A, UB2. We have like basically a cell cycle and proliferation and cancer markers over here, which makes sense. And on the left, like PCAM1, SRPR1, things like that, we have more vascular and stromal markers, which make more sense in normal tissue, right? So just on a quick glance, it seems like we're doing a pretty good job of that. One last thing to do, you can look at the MA plot. It basically gives you an understanding, one more look at something the volcano hides, which is the relationship between the expression level and the fold change. And you can see as we have higher fold change, everything's sucked down once again, because we have more faith in the values, where they go up higher on the end. So you see this kind of funnel out, deal here. And this is what you expect, a nice funnel. You can read a little bit more about it here in the bottom, and it's just kind of showing how the shrinkage is working a little bit. But all the QC looks really good, I think, and you will notice if it's more jacked up than that. So now we're going to do just a quick expression heat map of those top genes. We're going to grab the 50 top differentially expressed genes, and this time I'm just going to do it by adjusted p-value, and we will go ahead and find those in the VST matrix, and go ahead and just plot a nice, just simple clustered heat map of those. And what jumps out here, we see normal. First, we see normal tissue, primary tissue. We see upregulation of these genes here, and downregulation of these genes here. And we see the opposite in the tumor. So on the bottom here, we see downregulation of these genes, right? But upregulation in these tumors. Well, what kind of genes do we have here? Well, if you look in close, top 2A, KIF4A, we see cell cycle and proliferation genes. So that makes sense. Low, high in tumor, low in normal, right? The opposite is seen up here. We see more kind of vascular, stromal. What else do we got up there? Vascular stromal up here, which makes sense more in normal, and they're more lowered in the tumor, which makes sense because they're trying to pull some EMT nonsense or something. So this just kind of overall kind of shows things our top genes seem to make sense and plot out. So now that we've looked at that a little bit, let's take it a little bit further and find out. That's great what those genes are, but what biological processes are the genes involved in? So we can do these several different ways. We kind of build up here, and I'll show you in complexity. So first we do a gene ontology overrepresentation analysis, ORA. This is probably the least useful one to do, but it's a good first step to kind of take a look at things. And what I'm doing here is I'm going to find all my I'm going to basically take all my genes that are significantly upregulated up and down and create a background of all those genes. So when you're doing ORA, you need to always set a background just of your total genes. Otherwise, you're comparing against all genes, and you're trying to see if they're overrepresented in those genes. If you try more genes, you might get them in the wrong genes, or it just dilutes the value. Obviously, they can't be in everything, right? So you got to be able to balance that the right way. So we have 2,979 upregulated, 1,700 down, and our background universe is 1,699. So let's go ahead and do our Go enrichment, and we're going to use biological processes. You could also do cell components or molecular function, but BP is a good one, I think, to look at. We're doing our Benjamin Hochberg PA adjustment method with a PA cutoff of 0.05. And basically I'm just taking my signature up ones and running enrich Go with that universal background and just our simple, library-based human scaffold, right? And what we see here's our answers here. And kind of interesting here, we see that the top terms, organelle fission, nuclear division, chromosome segregation, those are kind of cancer proliferation hallmarks to be expected, right? So that's good, right? So we feel pretty good about that. There's a couple other things that are mixed, but, it looks pretty good. Let's look at the down-regulated ones now. What do we see? Blood circulation, wound healing, muscle system progress, reorganization, cellular movement, These are all things that are what normal tissues do, right? Vascular biology, the circulation, vascular development, endothelial proliferation, the cilium movement. These are all things that make sense in more of a normal tumor. So these are kind of making sense to going up and down, what we're seeing. Now let's do KEGG pathway enrichment. So once again, the sig up and sig down in background, but we're changing them to the intreg signals because KEGG likes intregs, right? And then we're going to do enrich KEGG and our same deal, enrich up and down. And then we get our KEGG pathways here. They kind of give us interesting up and downs there. And if you look at the top one, cell cycle is up there, and it looks good. We also have some weird ones up here like alkalolism, but that happens in KEGG. It doesn't throw off the ones that look good there. The downregulate, the bottom one's a little more interpretable. You have cytokine receptor interaction, chemokine signaling, but you also have vascular smooth muscle concentration, calcium signaling. So you see kind of a little bit of a mix, but it kind of leans more toward, a normal aspect, right? Down-regulated in tumor, upregulated in normal. So those are kind of looking like they kind of make sense. Our differential genes kind of made sense. Those guys kind of made sense. A little bit more statistics, right? It has to be statistically in there. But the problem with that is you had a cutoff, right? Sig up, sig down. You had this cutoff, and you didn't really be able to use all your genes. So one thing you can do to kind of pump up the statistical capacity and power of the analysis is move up to gene set enrichment analysis, right? So this gives you an advantage because it allows you to use all the genes, and they're also ranked by their fold change. So we don't care as much about the p-value, we're just ranking by their fold change, right? And this is kind of the modern gold standard for enrichment analysis. So basically, there's no cutoff, and I'm just ranking all the genes by log fold change and putting them all in. I basically created my ranked gene set. I grabbed my hallmark genes here, and then I ran FGCA. You see my hallmark sets I downloaded, my gene rigs I ordered, and then I just some default values here, and then we tidied the results, and here's what we get. And it's kind of interesting values here. We see things like EFTT, GTM checkpoint up high, TGF, TNFA, inflammatory down. So let's go ahead and plot these. They're kind of interesting here. See what they are. And this gives us a good plot here. And what jumps out of us here? Well, the red bars over here are pathways activated in tumors, right? So we have ETF targets. We have GTM checkpoint. We also saw those in Go and KEGG. That's good. We have MYC targets, which reflect oncogenic transcriptic amplification. We have glycolysis, in the Warburg effect. I mean, things are going really well. Even KRAS signaling down makes sense there. So that makes a lot of sense. And the blue ones are even more informative. We have TGF beta, which is which is only disrupted as it's disruptive as its tumor structural role is disruptive. We have myogenesis, which is more of that smooth muscle vascular thing we've seen in the Go results. And we're really just seeing more of the normal results that kind of back up what we see. So really, we're seeing the best thing about this is every step of the way we're seeing similar things that overlap, and that's what you want to see. You cannot trust each method. You got to go each one. So just kind of the enrichment plot's pretty cool. You basically just do a plot enrichment, hallmark sets, and your top pathway gene you want to do. So we're using the hallmark here. And you can see here what the black lines are just the genes in that pathway. So we're looking at the ETF targets, right? ETF pathway. So all these genes with black represent ETF targets, and we're seeing more on the higher ranked here. So we have a higher signal on the higher ranked side, so we know it's enriched there. So we feel good about that. Just a more proof that is going on. So what's the next step? Well, now we know what the genes are, we know what they're doing, kind of, what pathways they're involved with, but we don't know really, we haven't really been able to plot the per sample pathways, right? What is happening how do you look at the pathways per sample as far as clustering and activity score? So instead of saying, hey, is this pathway enriched? We're saying, how is this active pathway enriched in each individual sample, okay? So the first thing we want to do is get the hallmark gene sets, right? So we grab those here, we match them to the VST matrix, and we basically just run GSV. So we have our matrix, our clean matrix. We just kind of cleaned up how it looked. We add the hallmark ensemble, which is just the hallmark gene sets, and we're kind of just running it at default values, and we get our scores. We're going to go and plot these up. Just to give a quick overview of how this could work. So we have our samples on the bottom, and then we have our pathways here. So we can really jump in and see our normal here is clustered decently well, but there's kind of weird things going on with it a little bit. And then we see up here our primary tumor. So if we take a look at this, a few things jump out at us. But real quick here, this is GSV really bridges the enrichment analysis and clinical data, right? Unlike the gene set enrichment analysis, which compares group averages, this gives you a patient score for every pathway, right? And this just gives you things you can correlate with other things like stage, survival, mutations. It gives you a score, right? A pathway score per patient. So let's just kind of take a look at what we see here, right? The bottom block here, we have ETF, GTM. That's really a proliferation, cell we saw before. That cell cycle and proliferation. The top block has interferon responses, anti-inflammatory. It just seems very immune response going on up there. And then we see some kind of heterogeneity here in the middle with the EMT, angiogenesis, TGF beta, which doesn't a very good job of splitting the tumor. So these guys split the tumor well. These guys are kind of on the gradient between the tumors. So just if you get a little bit closer, you can cut this out to some of the ones that are more the ones that you're really going for, and give you a little nicer heat map there. But this is a really, really great way to get your per pathway score. All right, so the next and final step to really look through these different gene programs is look at the transcriptional analysis of this. Now, I remember we went through in our DNA section the non-negative matrix factorization and its ability to really, deconvolute the different signals within the actual expression matrix, right? And it did it by using a non-negative factorization, so it did not allow for negative values, which a lot of other methods do. So that really keeps the biology more realistic, and it really helps you decompose your expression matrix into actual small programs that are, metagenes, where each program represents a set of co-expressed genes that gets a weight to it, right? So it's a really, really awesome way to, as we saw with the mutation signatures, to kind of decompose the expression matrix and be able to find these different kind of programs. All right. So in theory, we should be probably looking for the three, like the true, PP, and PI. But let's see what we get. We don't know. We're going to jump in and try it out, right? And this is really going to tell us if we can get these transcriptional subtypes in our data. So let's take a jump in here. First, we're just going to prepare the data for MRF, because in MF, we just want the primary tumors. We're only taking the tumor samples. We're just grabbing the normalized counts from DDS, from DSeq, and then we're just finding our 5,000 most variable genes, and we're set there. So then all we got to do then is our final step, and that is to basically set up the non-zeros, right? So there's two ways to do this. Right now, this data, as I can tell you, this MF input data is in a Poisson or also possibly a negative binomial distribution. Now, as we do our NF methods, there's two NF methods that handle that well. So that's what the data is right now. So if you want to keep it like that, then we just add a +1 because it can't handle zeros. Now, that being said, there are other methods that want a more normal distribution, a more Gaussian. If you want to use those methods, you need to do a log 2 NMF input but 1 here to get that normal distribution. And we'll see why in a minute, why you'd want to use the different ones there. So anyway, this gets us our full NMF input to go right there. So next step, optimal rank. So this is really important. What's your K value? How many clusters? If you get this wrong, it throws off the entire analysis. So it's very, very, very important to either gain or lose structure. It's important to get this right. This is it takes forever to do. So I set this up as a parallel operation here. Basically, I am looking at K between 2 and 8, and I'm basically running this NMF estimate rank. I'm using the brunette method, which is just the Poisson method because we left it at the Poisson, right? We're using the NMF input, not the logged. And then we're doing 10 different runs for each. We're setting our seed. These guys just make it run parallel, and we're running this. It's going to run 10 times, 2, 3, 4, 5, 6, 7, 8, and give us some parameters so we can choose the best values. And this takes a long time to run, even with parallelized. Just let that know. So we've seen some of these before. Cophonetic, we want it to be high. Grab it before it falls down. Dispersion the same. Residuals normally go there. RSS, elbow, silhouette, we want to go highest before it curves down. So we've kind of seen some of these before. We know what to expect. So we kind of look at these and just see what we think is the best. And when I look at it, 4 seems good. 4 seems good. 4 seems good. So 4 to me is jumping out as being pretty solid among these. So mathematically looking at that stuff, I see 4. So what could that represent? Well, we could have 3 subtypes like we expect. And a 4th one could be something like tumor microenvironment, something like that. So no one knows the right number. That's why we're scientists, right? We're trying to figure it out. But this is a good way of mathematically to decide which one we want to use. Now, what methods will we use and why? So like I mentioned before, if you want to use the Poisson negative binomial method, you have to use brunette or NS-NMF. And brunette's very standard. The Lee one was the original NMF, but the brunette was the original NMF for RNA-seq data. And it does a pretty good job with count type data, right? So this is probably the best one to start with your Poisson's already there and your root method. If you want a Gaussian method, you do the log transformation, and Lee would be a good alternative as well too. If you want to kind of keep the brunette styling, but kind of go down and look for more, overlapped, harder to find boundaries, you want to go to this, start looking at the smoothing matrix to promote sparsity and to be able to find these smaller different things, right? Likewise, you could also take Lee down and use SNMFL, and this requires normal Gaussian distribution as well, and it basically forces the sparsity on the W matrix. And this is what you want programs defined by a very few highly specific markers. To me, brunette, Lee, and NMF1, these 3 are the best ones I've found to work with. But this is highly data-driven and parameter-driven. But just to give you an idea of which ones to use and why we would use them. And this kind of goes into that a little bit more of what you would do. Sometimes if you want to make it sparser, you can be able to find these harder to find things. All right, so now that we've found the rank, we've chosen the rank before, we're just going to run this again. This time just put NMF, rank K, same values below. This time we'll do 30 runs to get a little more iterative process. Everything else the same. We pull out our W and H matrices just like before with our DNA stuff. Remember that gives you your different, W is genes times programs, H is programs times samples. And then we're going to take a look at the H to find these programs, and we're going to see how many samples are in each of our programs and go in and plot those for us here. And then we also see the silhouette values here too, where the breakup is. Now, if I would have run this one time, it would have been super clean, all red and blue. But since we iterated it so many times, we saw the 30, you would do something like 100, 200 for your publication. So that means that every time it tries to put samples in a cluster, sometimes it gets a little bit wrong. So the more little white areas on there just shows more of the variability. But you can see they're pretty close. And if we look at our basis and consensus plots, they match up pretty well here, where we have, our different clusters here. So it'll be interesting to jump in and look a little farther into what these clusters, actually represent. And this is basically the consensus math to show this. So let's go in and try to do our little interpretation. We're just going to take out the genes, the top genes most associated, 15 genes associated with each program, and plot them out here by descending order of their weight to the program. And we'll kind of just jump out here right away and see what we think. Program 4 is a clean hit with all its surfactant proteins, and it looks like the true subtype, one of those subtypes we were looking for. And if we see this, I forget which one it actually is for, right? It's the plot right here, right? Because there's no gray on those, right? I'm trying to remember which one's which. The basis, because those are all the orange. Okay, remember now. So it's the basis plot. So on top, so the orange one's here. That's that true value. Sorry, the pink one is the right there is the true value, the one you expect to see, the true one we found. What else do you see? Program 2, lots of microenvironment there. The FN1, Col3A, Spark. So 2 looks pretty solid too. So 2 up here, this green one. So this looks like a good immune compartment. This looks like a good true subtype compartment that we feel pretty good about. But 1 and 3 are not doing too great. 1 already has some mixed signals that are in other ones, and it seems to have a lot of highly expressed housekeeping genes. It doesn't quite get you the PP program it tries to. In the bottom ones, you see a lot of mitochondrial genes. So it's possible the housekeeping genes, the mitochondrial genes, is push these genes expression down and squish them down to where you might find them a little bit lower, but it's kind of caused it to not get exactly the right MF. And this is exactly what we want to see in the teaching moment. If this all worked out perfect and they all were great, then it would be a joke and we wouldn't have to go forward. But this is what happens in real life. So what do we do now? Well, we probably want to go out and maybe regress out our mitochondrial signal at the very beginning of our data, or take out, filter out those values, filter out some of those genes, basically just get rid of the genes that have MTs on them. And then their signal wouldn't be there, and then you would be able to find a better, you could probably grab some housekeeping genes, the mitochondrial genes, ribosomal genes, take those out at the beginning, rerun everything, and you probably would get a more clean result. And that would be the next step to be able to do that. And you see here, when we plot them here, 2 and 4 were the ones that split the best, to be expected because they had the types, where 2 and 3 are kind of more intermixed. You can see how well these PCAs split these NMFs apart. It would be really cool to go back around and see it once again done, the correct way. So now let's jump to bulk deconvolution, which is really cool. People talk about cell types, deconvolution, and single cell, but you can also do it in a bulk sample, and it works pretty good. So remember, bulk RNA-seq gives you the average expression across all cells in a sample, right? But tumors are heterogeneous, and they contain lots of different cell types. So deconvolution algorithms can estimate the proportions of these cell types in your system, right? If you have more immune cells, macrophages, fibroblasts, Tregs, this is really going to matter clinically how you would want to treat that patient. So we're going to do a program called MCP Counter, and it does a pretty good job of doing this. And it was that DevTools program that we installed at the very beginning. And here we're just going to take our gene info, because we just care about our genes right here, to get our symbol map here. And that'll just give us our inputs, right, of our genes. And then based on our input of our genes, it is going to go back and look at the expression levels using this immune deconvolute package, and that will give us our results. So basically you see how we found the gene names, and then we use our TPM matrix. Remember, this is cross-gene absolute expression. This is the one place TPM really shines. So we used our TPM matrix from before, and then just chose our genes we want to look for in them. And then we just put that in there and ran it, and here are our results. And this is relative results. Don't be like, don't report in your paper, this has a 10 log PC. No, it's relative in what matters. So we know that fibroblasts were a lot higher, counts of those than NK cells. And there's also, see the spreads. So like, and this is just things we expect to see, right? Fibroblasts dominate. We see that in stroma heavy Luad. We have the same COL1A FN1 signal that drove it in ProVen2. Monocytes, macrophages, and dendritic cells kind of have that next tier, which makes sense, that myeloid compartment. NK are normally sparse, that makes sense. And what's cool is the T cells have this huge range, right? The T cell CD8 plus and cytotoxicity score, they have this huge range where there could be outliers here that are more immune hot that could have more benefit from immunotherapy. Something to think about. And we just look at this by stage. Let's look at just immune and fibroblast by stage. And we see some common things where you see B cells, T cells, they show a downward trend from stage 1 to stage 4, which is consistent with this progressive immune evasion during cancer progression. And you look at it two ways. You know, the higher the stage, also the harder for the immune systems to get in. These patterns are suggestive for some of the stage things, but this gives us kind of a good formal view. We need to look at it a little bit more statistically and hopefully single cell, but it just kind of gives us an understanding of things like, you know, the macrophages didn't change much because bulk can't split up M1 by M2. So that's an example of how single cell will make it better. All right, so the last thing we're going to do is survival analysis. So we're going to link our expression to our actual outcome. So doing survival analysis on mutations is more of a classical way to do it, but you can also do it in expression as well. Sometimes it doesn't work as well, but you can do anything with your survival. So let's just check out what happens and see if does expression of a certain gene or pathway or cell type proportion predicts survival. So we're going to grab just kind of tidy up our survival data that we have. You can kind of check out how I did that. And we end up having 393 patients, 129 deaths. The median follow-up is 21 months. And let's go in and look at this let's look at by stage first. So we look at just stage simple survival data, set up our survive fit on our OS months. So you look at the modes, events, looking at just stage simple. For the rest, I'll just kind of go through them. And you see by stage that the higher stage opposed to the highest stage is way significantly lower survival. Let's look at by gene expression. We'll take a few genes that we know to be popular, that have a lot to do with Luad oncogenicity. And we see that PV3 is not significant, EGFR is not significant, KRAS is not significant, STK111 is not significant. So what are the driver genes aren't significant in DNA expression? How could that be? What about MKI67? Well, that is significant, right? Well, that just means more proliferation and more cell cycle and more proliferation. Obviously, that's going to lead to worse outcome. That makes sense to be correlated. But what about all of our oncogenes? How are they all non-significant? I thought they were driving the cancer. But that's not a failure of the analysis. It's real biology, right? These genes drive cancer through mutations that alter protein function, not through expression levels, right? So a missense mutation in TP53 could produce a broken protein at normal mRNA levels, right? Or a KRAS could inactivate signaling without changing transcriptons. So that's why you need DNA, copy number, and RNA to be able to find these things. So it's just an example of how a lot of time oncogenes don't fit in. So let's look at other things that actually could fit in, right? It could form scores. A lot of times scores and combinations do a better job of predicting survival. So let's jump in and look at the NMF clusters. Let's see if those do survival. And we look right away, it does. 1 has the worst survival, and 4 has the best. So what is 1 and 4? Let's go back. It's kind of weird, right? So 1 and 4. So 4 had the worst, right? Well, that was the real subtype, right? Or 1 had the highest, where it was more of the housekeeping gene phenotype. So let's look back at this and see what we get. It's kind of interesting. So the worst survival was that 1, housekeeping. Oh, the best was that true subtype. That's right. But we do see some split here, right? So despite the imperfect decomposition, these programs actually do predict survival pretty well. With 4, true, the one we actually got has the best prognosis, which that is what it should be. It's the least aggressive, right? So program 1 is the worst. We only had a few patients, so it was kind of weird. But we're just kind of seeing that even with our M&F not perfect, it is still separating out things that make biological sense. So we just need to kind of correct that, go back, refilter, and redo that. I think we get a better result. Let's look at the GCVA pathway score. This is a really cool thing. We can look at pathway scores. So we chose the EMT here, but we could have chosen any of the pathways. And let's do survival on that. Well, we see EMT pathway does have a worse survival. So that's cool. We could try all those pathways and find some really cool ones. And this makes total sense because it's consistent with EMT and invasion, metastasis, and treatment resistance. So this teaches us more about what's happening in our LUAD sample. All right, so that just shows how survival can handle all those things. So wow, I think we've had a great lesson today. I can't believe all the great things we've covered. And I'll kind of leave this for you guys, kind of just summary, kind of a decision tree on kind of how to go through your RNA-seq analysis, different expression methods, different enrichment methods, deep convolution methods, everything you can possibly think of to go through there. So, as always, now we go through the main lesson. Let's go back through a few practice problems and hope these can kind of dive in a little further and add some value and context to what we've already gone through before. But I also won't go too in-depth because I want you to kind of go through them yourself too, because that's the way you learn. So let's first just do a very similar thing that we did before, but with the kidney cancer data set. Let's dive in. I've never seen this data set in my life. Let's take a look at it and see what we get. So we're basically just going to download the TCGA kidney cancer, and things we're going to be looking for or different pathways that are driven. This VHL pathway really drives this kidney renal clear cell carcinoma. So we're really going to look at how VHL destabilizes HF1A2. These are hypoxia-inducing factors. So it's very hypoxic related, even under normal oxygen levels. And it's also going to have downstream effects with angiogenesis, glycolysis, and cell proliferation. So these are the things we're going to look for. Will we find them? I don't know, but we're going to look for them. And here's some more of kind of the things that we're going to be looking for. So part A, we're just going to download the star counts exactly like above, nothing different. You can check it out above to see the same thing. We have our sample types with a good number of solid tumor tissue. And this time we can filter by 72 for the low instead of 59. And just like before, we're going to take out the protein coding genes, remove the low expression genes, and this time we'll do one step further, and we're going to remove the ribosomal, mitochondrial, and housekeeping, just for fun, to show you how you do it, so you can use this code up above if you want to redo it there. So these are the housekeeping genes, these are the mitochondrial, and these are the RNA level. The ribosomal genes. Lose my mind. And you see the kind of the filtering here. We filtered down a little bit down on that. Hopefully that'll clean up some of our downstream analysis. Then we do a DSeq data set with our counts. And once again, by sample type, this is all exactly the same. We do a VST as well. And then we just do a PCA on that. We see good separation, a couple little overlap that could be interesting biology. And then we jump into our differential gene expression, DSeq on our object we built. And then we do our LLFC shrink for the ape shrinkage, right? HTLMM, and we copy this, see how many genes we have, and then we look, we're basically going to subset out just for these VHL HIF pathway genes and see if they're differentially expressed. So we just grab those genes. They all are significant here with decent. Most of them have decent log fold chains values, and you see they're up and down as far as direction in the tumor. And what jumps off right away is this VHL HIF axis is just unmistakable. You have K9, VEGF-A, SLC2A1. These are strongly upregulated with a full suite of HIF target genes, e.g. LN3, ENO2, PD. I mean, this is just nailing everything, right? Exactly the kind of things we would expect. So VHL isn't, but VHL is a little bit down, but it doesn't matter because all these other things are taking, it's not showing up in its expression, it's showing up in everything and everything else. So we're really kind of seeing what we expect. So do we see these? The idea is, are we going to see this hypoxia and glycolysis light up if we go a little further in the analysis? That's what we'd expect. So let's build our ranked gene list, and we're going to do our gene set enrichment analysis. So we just ran that again with our ranked genes. We skipped the ORA, and we take a look at this, and we once again see kind of immune dominance on the top there, but we don't see this hypoxia and glycolysis like we expect. But what we do see is a bunch of immune pathways like allograft rejection, interferon gamma response, inflammatory response, all this stuff. It's kind of pushing down what the things we really want here. But the VHL, the axis is still there. Look at the negative values, right? Look at these negative values. We're seeing oxidative phosphorylation is negative. What else is negative? Fatty acid metabolism is negative. There's other things that show that this pathway is in there. So we're really signaling. If you want to look into a little bit more, you'll see that. And you can see it plotted a little bit here too, where we see hypoxia is pulled way down, but it's well behind all these immune signatures. And glycolysis doesn't even make it, but it is there. But what does this mean? Is it not active? Well, on the bottom, you see the very suppressed pathway showing it is active, but in reverse, right? So the immune pathways on top are having a dominating effect that we're losing out on the signal, but it doesn't mean the signal's not there. It's just being squished down a little bit, right? They're still there, that's significant. So let's look at the enrichment scores. We'll look at the hypoxia enrichment score. It's very good. The glycolysis is really good. So hey, they're really good. Don't mistake being outranked for weak. The signals are great. They're just outranked by all the other stuff. So we are seeing what we expect to see there, and we feel really good about that. We could also go into see about how about the survival of this expression. Well, we're talking enough to be so important. Does survival matter? Nah, it doesn't. While KA9 had the highest log-fold 2 change in our entire analysis, it's a direct HIF target, and it's used clinically to diagnose. So you expect to see it to predict survival, but it doesn't. But this is a really, really important lesson, I think, that statistical significance does not guarantee a prognostic value. And while it's massively upregulated, it's nearly regulated in all the tumors. You can't really, it's a very universal subtype. So you can't really use it to split up different patients to find different survivals, right? So it's really hard to split up. Doesn't mean it's not important, it's just hard to split up like that. All right, so problem number 2, the other gold standard, Lima, DSeq versus Lima-Bloom. There's a whole slew of people that like to use DSeq or Lima. DSeq's kind of used it for simpler things, where Lima is used for more difficult things, I think. So the difference is the Lima uses a linear model on log CPM instead of this negative binomial GLM. Its normalization is from the TMM, if you remember, instead of the MLR. It uses precision weights instead of dispersion, very similar, a little bit faster. It's really better for large sample sizes and very complex designs, like adding a lot of different interactions, things like that. So we're going to use the TMM normalization that we already did up before. So we already taken that in. It already has the weights. So we'll use those objects from before. So we have our counts and our sample types. And then we're basically just going to recalculate those TMM just for fun. And then we have our TMM values. So once we do that, we build this model matrix. And basically what you do here is you have 2 rows, 1 and 2, basically your different things you're comparing. And then you have a 0 to 1. So you have the reference is 1, right? Let's see. How does this work again? So a 1 means the sample belongs to that group. 0 means it doesn't. So for each sample, we just say, so if you imagine every row, if it's normal, it'll have a 1. If it's primary, it'll have a 0. And this kind of goes all the way through, and it creates this matrix. And this is the way, instead of having the row of sample type, this is its way of knowing which is in which. It's kind of confusing, but you basically just, it's not that hard. You basically just make it right here. You put a 0 in, you put your sample type, and you put your data. Model.matrix, it made it. Design. Bam. You don't have to worry about that anymore. And then you go ahead and can throw it in here, and you have that DJ. Then you want to basically do your input Then put the VUM as your DGL list, which you've already done and calculated before with TMM and all that. So we will go ahead and put that in. The CPM, if you remember, on that gives us our final value. And then the last step is to run that through VUM. So that gives us our value here before VUM. This is just our TMM, just like before. And now we do VUM, one more step, we VUM that value. And this converts the counts to log CPM and estimates proficient weights. And it helps make this little guy just like very similar to what we saw in DSeq before. So you expect these values to be shrunken in more to the model. That's how you can kind of tell how well that works. So really it's the same thing. We did our TMM just like above. We made our design matrix right there. And then we just ran our CPM just like above, got our result. And then the extra step we're taking is doing our VUM. That gives us our final pretty much value. And we can see what VUM did, pretty much nothing because it's just computing the precision weights. It's not changing that. You can see here, the weights get higher as the counts get higher because you trust those guys more. And you can kind of see the weights here. But long story short, you take that VUM result, put in LM fit here with V and that design matrix we made. That gives you your fit. Then you make your contrast, just define the contrast, and you apply that contrast. And then you finally do one more step. You do this empirical Bayes method that shrinks everything down kind of like that ape GLM. And then you just extract your final results here. And then if you look here, you can look at your differential genes just like before and get your top 20 differential genes, things like that. And if you notice here, it's kind of interesting that the top 20 genes are all down-regulated, right? Genes like FAB-P4 and COLEC3B are expressed in alveolar endothelial cells. They get displaced as the tumor grows. So this kind of makes sense when DSeq Heimer tumor gains, Lima's Heimer tumor losses. And you can see here, it's really focusing more on the tumor losses than the gains. But it's just interesting that it has different results. That's the most important thing, right? So let's compare all the different genes we found in each of those. If you compare the genes, of DSeq versus Lima, we see that Pearson's a little off. It should be around this red line, but it's a little off. But 0.837 is actually pretty good. So it did a pretty good job of agreement. It's not perfect, but what you will see are things like, look at where the blue regression line separates from the red line. For large fold changes, Lima constantly estimates smaller effect sizes, right? So it's doing smaller, while larger ones is doing bigger. So they're just kind of, it matters. Their shrink methods work a little bit different ways. And kind of just comparing them here, we see that that there's only about 11% that were found in one or the other, and 20% were found in both. It's really out of 40%, 33% here. It's knocking out 21% in both. So that's pretty good. So what's happening to these? Are they just completely wrong, or is this a matter of like just significance, right? And if we plot these on this plot, what we'll see here is with the scatter of the fold changes is here they're both significant, here they're both significant, and here are the differences, but we don't see anything here, right? So what this is telling us is that the discordant genes reveal the shrinkage difference we saw earlier, right? The red dots only cluster right below the diagonal, where the DSeq preserves the larger fold changes that the Lima compresses below the threshold, where the blue dots on the downregulated side are actually with the small fold changes. So what's interesting is almost no dots fall in opposite quadrants, right? So that tells you, that's really, 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 really important. So that tells us that the methods agree on the direction almost every time. So they're agreeing up and down. There's just the different ones that are only one or the other, they're just probably very like cutoffs for fold changes or cutoffs for p-values, things like that. So they're a lot closer than you think probably if you really read into it. And then let's just jump on to our problem number 3, kind of finish this day off. And this is kind of a cool, just advanced little project I made. So I'm always trying to get you guys just to develop your own like novel risk scores, things like that, prediction models. Well, here we kind of do both, where we build a multi-gene prognostic risk factor stores are really simple, but kind of cool. So the first thing we want to do, so multi-gene signatures are very important, right? These are being used in clinical assays and clinical trials, therapies, to really do a good job of helping disease now. And what we want to do is basically find a way to look at the expression of a bunch of genes and make a risk score for LUAD. So how can we do this? So first we're going to select some candidate genes, which in our case are going to be the top differential expression genes they've already calculated. Then we're going to fit really a univariate Cox model first on every single gene and just find the ones that actually predict survival. So we'll find all the differential genes, then we'll look at the ones that just affect survival, then we'll build a multivariate Cox model to really plot the ones that really matter, they're independently predictive, and then we'll build a risk score, and we'll talk about that later, and then we'll validate that with actual survival data and test the independence too. So this is actually really cool. So for select candidate genes, I actually grabbed I say 100 here, but there's no point in not grabbing a lot, so I grabbed 1,000. I really wanted to find a lot of genes that I can find out, but you can try any number you want and see the different answers you get. So I grab 1,000 possible genes. And then I make a loop here, and all I do is do a univariate test each time. And I'm just saying, hey, just you alone, what's your hazard ratio? Do you have a hazard ratio below 1, greater than 1? And what is the p-value on that? So I did that for all the genes, and I found that 293 are statistically negatively affecting survival. And I pulled them up here, the top 20 of those, by the C index, which kind of ranks how well the p-value and the HR value affect direction. And you can see that, remember, over 1 is bad, under 1's good. And you see here that we have a lot of genes that were highly differentially expressed, but also affect the tumor survival. So now we have kind of a list of cool genes. We're going to take all those genes. We're going to take the top 10 of those and make a multivariate model and see how that works out. And when we do that, we see that actually only 3 indups that are actually significant now. So we take those 3 genes. One has a high log fold, one has a lower. So 3 genes basically survive this. And they capture the most value of it. But I'm actually going to use all 10 of the genes for this. Here's kind of the distribution in the risk score here just to show you that it's a kind of a normal distribution, which feels pretty good about. And let's talk about the risk score. So basically all I'm going to do is very simple. I'm going to multiply the hazard ratio times the expression and then do a linear combination for the whole sample, right? So here you go here. We get the Cox coefficients we calculated earlier, basically just looking at the log HRs, right? And then we're going to multiply the expression times the coefficient, right? So the Cox coefficients, the log hazard ratio values times the expression will give you your risk score. So then everybody has its own risk score, right? So we look at, and it's basically the risk, my patient has a risk score. Now we're going to split these by the median to high and low. And we see here a high risk, and they're both negative actually, but that's okay. It's the relative position. So the higher the value, the more risky. And then we're going to go ahead and throw that in a survival analysis. And wow, there's a pretty significant difference. A much worse survival with a pretty balanced groups with our scores. It actually worked really well. So it just shows you the value of some really simple little thing you could throw on a paper and publish. But one critical caveat here, we trained the model and evaluated its performance on the same patient. We really need, in a real study, you want to train on one cohort and validate it on a completely independent data set. Otherwise, there could be risk of multi-gene risk score not being clinical deployable. So let's take this with a grain of salt, but it's a pretty cool concept, and that's all really what I'm trying to do is giving you proof of concept, the things to do, giving you ideas of how to add extra cool stuff to your data. So finally, is this risk score independent of stage and age? So we run another Cox multivariate model on it, and we see that risk score is consistent, significant. If we look here, we see that even based on stage and age, risk score is significantly higher, and not only is the ratio much higher, the hazard ratio much higher, it is also the only significant one. All right, so it just kind of shows you we make a cool score. Here's a little bit we learned in our practice problems, some common things to avoid, some additional resources and papers you can go through to learn. I always love the stat quest, but you can really look through this DeepSeek walkthrough. But like I said, I think going through this is almost better. Here's a little bit of hands-on code practice and some things you can look through to find that data. And that's it, I guess. Thank you for being a part of this thing on this long journey on the bulk RNA-seq. So I think we've mastered this. So now next workshop, we're moving from bulk to single cell RNA, where we can have these same questions, but resolve them at the single cell level to understand things like cell types, cell programs, differential expression at the resolution of individual cells. So we'll be able to do dimensionality reduction, graph-based clustering, cell type annotation, pseudo-bulk differential expression, and cell state scoring. That'll be our first half. So the first half is going to go from counts to annotations, and the second half is going to be more subclustering, downstream analysis like cell chat, trajectory analysis. It's going to be really cool. I think you guys are going to learn a lot, and with real world data, not some trick stuff. So I guess that is pretty much it for today. Thank you so much for joining me once again, and we will see you in the next workshop. Thank you.